session, we are going to discuss about transfer RNA as an adapter molecule based on your molecular basis of inheritance of your textbook. As we all know that the second part of our central dogma of molecular biology, the translation in protein synthesis, we use three types of RNAs. Messenger RNA, which code for the amino acid. Then comes the transfer RNA, which can carry the amino acid from cytoplasm to the site of protein synthesis, and the ribosomal RNA, which constitute the structural part of the ribosome. Today we are going to discuss about the structure and the different organization of transfer RNA. As we all know that this transfer RNA can carry amino acid towards the ribosome. When we look at the details about these transfer RNA, we can see that these properties are prescribed by 3S. This is the smallest type of RNA. Among all the three RNAs, transfer RNA acts like the smallest type of RNAs. Almost 75 to 95 nucleotides only are present in this particular RNA. That is why we can consider it as the smallest type of RNA. Then you can see that the RNA is of single strand that can start from 5' end and ends in 3' end. So we can see that this is in a single stretched runs. And this has got again the third character, that it is also called soluble RNA. Why it is called soluble RNA? Because 1 molar solution of sodium chloride, this RNA is highly soluble. That is the reason why we call it as soluble RNA. Now we will go for the structure of the transfer RNA. The most accepted structure of transfer RNA is postulated by Robert Hawley, and it is in 1965. As per this structure of cloverleaf model, we can see that this single-stranded RNA takes certain twist or coil itself. Because of this reason, the single-stranded RNA looks like a twisted and coiled structure. But the real three-dimensional form shows that this 5' which starts from this particular point and this end in that 3' end will take many turns. The first turn comes down and take a loop, that is the first loop, and then comes for the second loop, then comes the third loop and ends in the 3' end. Always the 3' end has got a free OH group, and moreover this 3' end is always carrying a specific sequence of nitrogen base CCA. So every transfer RNA at the 3' end, we will see that there is a free OH group as well as the nucleotide sequences as CCA. And when we will see the first loop, it is called as D loop. The D loop is technically consists of certain specialized binding site of a particular enzyme. The enzyme which is coming as a link is that amino acid transfer RNA synthetase enzyme. So there is a very critical enzyme which is needed for the initiation of the translation, that is amino acid transfer RNA synthetase enzyme. This enzyme is binding at this D loop. Now we come to the second loop, which is the bottom part. We can call this loop as anticodon. The term anticodon stands for which will get a corresponding complementarity with the codon on the messenger RNA. As we all know that messenger RNA has triplet codes which will represent amino acid. So each triplet code is carrying different amino acid one after the other. That triplet code is technically called codon. So when we see this codon, we can see this codon is in complementarity with the anticodon. So always this anticodon, anti means against. So this anticodon will be always coming as an opposite and the complementarity with the codon of messenger RNA. So this is the real site at which the messenger RNA is getting connected with the transfer RNA. Then we can see a small, very vestigial loop or a folding. This is also called a vestigial arm. The functional role is insignificant. There is no much significance in its role. Then we go for the third loop. The third loop is technically called T loop, and this T loop is mainly responsible for binding with the ribosome. And we can see at the 3' end, the amino acid, the activated amino acid can bind. So this is by the 3' end, the amino acid which are activated with ATP are coming and attaching. So with this amino acid, which is complementary to the codon of the messenger RNA, we have to give the specification that the codon is technically giving the information for bringing the amino acid. So this codon as well as this anticodon is complementary. So this codon will be always getting the same sequence with the amino acid which is brought towards the ribosome by this transfer RNA. Then Francis Crick has given a special position to this transfer RNA, that is, it can act like an adapter molecule. Now we are knowing what is the role of an adapter. When we use that electronic gadgets, we will see adapters are always helping in linking two devices. So the same way the adapter molecule transfer RNA can get a link between the codon of the messenger RNA with the corresponding amino acid. So always amino acids are attached to the transfer RNA as per the codon sequence. So here the transfer RNA is acting like an adapter molecule. So we are getting all the structural modification which is represented by clover leaf model. This clover leaf model proposed by Robert Hawley in 1965, and this structure is highly responsible for the process of translation. I hope that the structure of transfer RNA is clear for you, and this will enable for the process of translation. Thank you.